Happy Friday! It never fails to elicit a few woos, right? By the end of the term, it's kind of like, right? Now it's still the woo. Big weekend plans? Studying biochemistry. This is good. The heck with biochemistry, right? All right, so we're pretty much on target with where we need to be, and very happy with that, and very happy with the interactions I've had with many of you so far. So it's been very, very positive for me. Very, very much enjoyed that. We're turning our attention now, as I said last time, to thinking about using our knowledge of protein structure as a way of working with proteins. Biochemists are pretty nerdy people, and as I'll probably say further in the term, and probably next term as well, biochemists are also lazy people. So we like to find easy ways to do things, and that's one of the things that we work on pretty hard. So you'll hear about some of those later as I talk about some of the techniques. So we're turning our attention for a couple of lectures to techniques that we use in biochemistry, specifically to isolate large, and in some cases, small molecules. Okay? It's mostly relevant for the proteins we've been talking about, but we'll talk about some other things as well. All right. Well, the number one tool that you see biochemists use in a laboratory, you can't walk into a biochemistry laboratory without finding a centrifuge. Okay? So a centrifuge is something that is ubiquitous, and it's a tool that allows us to do a sort of gross separation on the basis of size. Gross meaning rough, okay? Not gross meaning bad. So having that separation technique is very useful. So for example, when we're working with proteins or molecules, metabolites, or any of these things. They're coming from out of cells. And if we want to isolate them from the cells, we have to do something about separating the various components of the cells, because separation is a means for isolating, or what I call purifying, a molecule. Okay? So if we want to purify a molecule, we really need to get that molecule separate from everything else. So what we will see in this process is that there are numerous steps that have to happen in order for us to be able to do that. You'll see there are some shortcuts that work fairly well. Other times those shortcuts don't work well. So there's no one way of isolating molecules. There's no one way of isolating molecules. So some molecules take a lot of steps. Some molecules don't take so many steps. And I'm hoping to acquaint you with some of the steps in this process. Well, as I said, centrifugation is a way that we start because we first of all have to get the material that is part of the cell. So the material that we're working with might be in the cytoplasm of the cell. The material we work with might be in the nucleus. It might be in the mitochondrion. Of course, if we're talking about eukaryotic cells. If we're talking about bacterial cells, then it's basically in the cytoplasm or it's in the membranes. And of course, it might be in membranes of eukaryotic cells as well. So we have to have ways of isolating these various components. And centrifugation turns out to be a really, really good way because these are very large complexes. And the way that we separate them is by what's called differential centrifugation. All right? So differential meaning that we use different rates of spinning the centrifuge, and those different rates of spinning will cause different things to go to the bottom of the tube. All right? So you can see here a depiction of some of those steps. We've taken a bunch of cells, and the first step in isolating anything is busting open the cells. There are numerous ways of busting open the cells. We can sonicate them. We can, in some cases, treat them with enzymes. In some cases, we might simply use high pressure. And this particular method right here involves mechanical agitation. It doesn't really matter. All right? First thing we do is bust open the cells. When we bust open the cells, we've got the contents of everything that's in that cell in that tube that we're starting with. You can see that here. If we spin that at a low rate, we will precipitate certain big things. We spin it at a higher rate, we'll precipitate smaller things. Okay? So usually we'll spin it at a slow rate at first to get rid of the cellular debris, the nuclei, and things like that. Okay? And then we'll pour off that supernatant and take that supernatant, which is the liquid part, and spin it at a higher rate. So then when we do that, we'll spin out other things like mitochondria, etc. Okay? So depending upon where our protein or molecules of interest are in the cell, we can isolate those components relatively efficiently. Okay. Well, let's assume that we've done that, and now we've got this soup of whatever it is that contains the molecules that we're interested in working with. Okay? So one of the very common techniques that we work with, and particularly it works very well with proteins, for example, and one that you probably did in basic biology or maybe even chemistry when you took things, was to use a technique called dialysis. Now, dialysis is a way of separating small things from big things, and the basis by which it works is it involves taking your soup. Your soup is the thing that you've taken out of the centrifuge that contains the material that you're interested in, and putting it into what's called a dialysis bag. So we've done that here. The yellow that you see on the screen is the soup that we have. Okay? And the dialysis bag has a very useful property that it is porous to the small molecules that are contained in the soup, but it's not permeable to the larger ones. So, for example, I might have a soup here that is full of salt, sodium chloride, or potassium chloride, or something like that, small molecules, and I want to get that protein and the other things that I have in here separate from those small molecules. Well, with dialysis, it's very simple. The small molecules, as I said, are permeable. They'll come out. The larger molecules are not permeable, and they will remain on the inside. Okay? And in doing this, I'm thus able to remove most of the salt away from the proteins or the other things that I'm interested in here. So dialysis turns out to be a very simple yet useful way of separating big from small. There are other ways of separating big from small that don't involve dialysis tubing, and they may involve separation of bigger molecules, for example, that wouldn't fit through a dialysis tubing. All right? This technique called gel filtration, which is also called molecular exclusion, gel filtration equals molecular exclusion, involves a very cool trick. Okay? Well, as you'll see in these various techniques that I'm going to show you, the materials that are doing the separation are typically used in what are called columns. And the material that does the separation is actually shown over here on the right. So it's sort of an enlargement of what's in there. And I'll tell you a little bit more about that. Okay? So let's imagine, first of all, let's talk about what the material is. All right? So let's say I've got a mixture of proteins, and I've got some proteins in there that have a molecular weight of about 5,000, which wouldn't fit through a dialysis tube. And I've got other proteins that have a molecular weight of, let's say, 80,000, which also wouldn't fit through a dialysis tube. All right? So I can't use dialysis to separate those. I've got to find some other way to separate those proteins from each other. Molecular exclusion, or gel filtration, and by the way, the word chromatography comes up. When I say gel filtration, chromatography, all the chromatography means is separation. Okay? So gel filtration, separation, molecular exclusion, separation. Okay? That's what those refer to. Now, these columns that I have here contain the material that allows the separation to occur. So as I said, I want to tell you a little bit about what these materials are. As you can see on the enlargement, these are little round things, and this is a pretty big enlargement. It turns out that these materials, as we use them in the laboratory, are little tiny beads. Okay? Little tiny beads, maybe a millimeter or less in diameter. They're pretty small. Okay? But they have a really cool property. The cool property that they have is that they have holes in them, and the holes are connected to tunnels within them, and the tunnels go in one place and come out the other place. So things can travel through these beads. Now, this technique relies on something very important. And the important thing that it relies on is the fact that the holes and the tunnels all have a fixed size. They all have a fixed size. That fixed size is called the exclusion limit. Exclusion limit. Why do I tell you that, and why is that important? Okay. Well, if they all have the same size, the exclusion is that they're only going to allow molecules of a certain size to fit into them and travel through them. So I gave you an example of I've got some proteins that have a size of 5,000 in molecular weight and some that have a size of 80,000 in molecular weight. If I used a set of beads that had an exclusion limit of, let's say, 30,000 molecular weight, what it would mean is that anything that has a molecular weight of less than 30,000 will fit in there, and anything that has a molecular weight greater than 30,000 will not fit in there. Everybody got that? So the exclusion limit determines which things will fit in there. Now, what does this mean? All right? Well, it turns out to be very simple because if we think about the small guys going in here, traveling through the column, what we start with is a mixture of proteins. We put them on the top of the column, and then we take buffer and we let buffer flow so that it's flowing through the column, it's flowing through the beads, and we're just simply monitoring the rate with which the proteins pass through there. Now, because one group of proteins is traveling through the beads, and the other group of proteins is not traveling through the beads, it means that the ones that are traveling through the beads are in fact taking a longer distance through the column. So therefore, things that are less than the exclusion limit will come out last. Things that are greater than the exclusion limit will come out first because they don't enter the beads at all. Question?
Those beads are in a fixed position. They're not flowing.
The beads are in a fixed position. That's correct. Okay? The beads are in a fixed position. So buffer is flowing through them. It's flowing through the column. But for the most part, we look at this thing right up here, we can see that the yellow guys, which are so big they don't fit in there, are zooming through. The green ones, which are close to the size of the beads, may fit in some cases, not fit in other cases, and they go in the middle. And then the little guys up here that are the size, less than the size of the exclusion limit, they travel through all kinds of beads. They take a much longer path going through. Gel filtration chromatography allows us to separate proteins on the basis of size. We'll see other ways of separating proteins on the basis of size as well. Yes, question? Yes. Her question is, there'd be more than one protein in the sample. That's a very important thing to recognize. Yes. Because if I only had one protein in the sample, I would already have it purified. Right? So I've got to have a mixture of proteins. And usually I won't even have 2 or 3 like are depicted here. I might have a few hundred. So this isn't a way of getting a protein pure, but it's one of the steps in getting a protein pure. Yes?
You'd only be able to separate them as a group.
Well, not completely. His question was, I can only separate them into 2 groups. I've sort of drawn this so that we can imagine they're coming off actually in 3 groups. And so that's basically what you'll see. You'll see a group that comes through fairly fast, a group that comes through less fast, and a group that comes through the slowest. Okay? So roughly 3 groups. Yes? Is it fairly clear to tell when you're transitioning from one to another? Only if you have colors like these. So what you do is you will collect a few drops in a tube, then you'll switch to another tube, switch to another tube, another tube, and then you'll use other analytical techniques to tell you what was in that tube. Good question. Good question. Yeah, Kimberly. Very good question. Does the structure of the protein change as it goes through the beads? Hopefully not. Hopefully not. But remember that structure of protein is very critical, so your question is very relevant. If the protein structure changes as it's going through the beads, we might end up with protein that's pure or relatively pure, but it's not active. So that's a consideration sometimes, and that's why the choice of the buffer that we use is very, very carefully done. Okay. So that's gel filtration, gel exclusion chromatography. I want to tell you about another technique that we use that actually relates to something we've been talking about in class so far. This is called ion exchange chromatography. And you can understand it pretty simply, all right? But I'll describe it to you. Ion exchange chromatography also uses little tiny beads, just like gel filtration chromatography, except the beads that we use here they do not have any holes in them. So no holes. Instead, the beads have molecules that are attached to the surface of them that have a charge. So they have molecules on the surface that have a charge. Now, wherever we have a charge, we have to have a counter ion. You can see this set of beads all has a charge of negative. When we started out with this bead, it wasn't negative. It had a sodium ion or a potassium ion that was a counter ion to it, so that its overall charge was 0, right? We always have to have a counter ion. We don't have these charges just by themselves when we pour something into a tube. So we had a counter ion. We had a sodium or we had a potassium that was positively charged. Okay? When I apply my mixture of proteins to this, I'm going to have proteins in there that some are going to have a positive charge, some are going to have a negative charge, and some are going to have a 0 charge, right? Okay? Well, if I have a positively charged protein, that positively charged protein can exchange with the counter ion. That is, it can replace a sodium or a potassium, and then that positively charged protein is going to stick to the bead. So the exchange is the critical thing. If we know what the counter ion is, we know the type of chromatography that we're doing. All right? So if the counter ion is a positively charged molecule, we are doing what's called cation exchange chromatography. This shows cation exchange chromatography. Now you might sit here and think, well, is it possible to have beads with a positive charge and then have a negative counter ion like a chloride or something like that? And the answer is yes. And if we had that, then we would be doing anion exchange chromatography. Now, I think you can pretty well figure out what's going to happen and what's going to be the difference between these two. If I'm doing cation exchange chromatography, the molecules that are going to travel the slowest through here are going to be the positively charged ones. The one that's going to travel the fastest through here are going to be the negatively charged ones. And the ones that are going to be in the middle are going to be somewhere in the middle of those charges. Yes? Why doesn't the protein interact with the charge that it's displacing? Well, it can to some extent, but remember that it is positively charged. There wouldn't be a reason for it to interact with a positively charged potassium. So if I were to ask you this question on an exam, you just made me think of a question for an exam, so I'll tell you. I'm going to tell you what the question is. I'm not going to put it on the exam necessarily, but it would be a good exam question, maybe a future year, right? All right. So the question is, where would you expect to see the counterions? Attached to these guys. Attached to these guys coming up. Because they're going to be positively charged, they're going to find the negatively charged proteins that are in there, and they're going to come off with them. Make sense? Do you want me to ask that question on the exam? No? I just told you the answer. Yes? Say that again. Okay. Her question was, how come the proteins don't interact with the counter ions? I said, well, these proteins aren't going to interact with the counter ions because they're positively charged, and the counter ions themselves are positively charged, right? Where would we expect to see the counter ions? Attached to the negative proteins that are coming out down here. These won't have counter ions. They're going to stick to the beads. These guys down here are going to stick to the counter ions. Maybe I can think of a harder way to ask that question, so we'll have it on the exam. How's that? Oh. Okay. Yes?
Is there anything to prevent us from using both kinds of chromatography in sequence?
Is there any way to use both kinds of chromatography in sequence? In fact, it turns out that people do use both, and they'll actually mix these beads together, and they make something known as, and you've heard of this before, a water filter. Water filters are really good at pulling out an awful lot of positive and negatively charged things and letting everything else pass through. So yes, very good question. Okay, so that's what's going on with these guys, and I think you can hopefully understand that fairly well. Okay, this just shows you the chemical structures of some of these things. You don't need to know them. I'm just showing them for your own information. There's the structure of a resin that would be used for cation chromatography. This would be used for anion chromatography. Remember, it's named for the counter ion. So the counter ion is positively charged. The counter ion here is negatively charged. Okay. Now, I'll tell you about a technique, another type of chromatography that is very powerful, and it's also very easy to understand. And whenever you can do this type of chromatography, you can usually reduce the number of steps in your purification process by a long ways. It's called affinity chromatography. Okay? Now, this technique also uses beads. But instead of having charges as the basis or having size as the basis, these guys rely on the fact that many proteins bind to other molecules. So, for example, we're going to talk about proteins later this term that bind to and use ATP. Okay? They bind to and use ATP. Well, let's imagine for a moment we take those beads that we've been talking about all these times, and instead of attaching something else to them, we attach ATP to them. So we've got a bead that's full of ATPs. And let's imagine that I take my mixture of proteins that I get from the cells, and I pour them on the top of the column, and I let them come out. What's going to come out first? Well, the things that are going to come out first are those proteins that don't bind to ATP. The things that are going to stick to the column, at least at first, are going to be the proteins that bind to ATP, because they're going to bind to the ATP on there. All of a sudden, I've just purified all of the ATP binding proteins in that mixture. That's really useful. Okay? Now, there might be a bunch of proteins that bind ATP, so I might not have it pure, but let's say I know of something that only my protein binds to. It only binds to Kevin Ahernium, right? And we attach Kevin Ahernium to this column, right? What's going to happen? I'm going to purify that one protein that binds to Kevin Ahernium. By the way, I don't want to interact with that protein. Okay? You see how powerful a technique this can be? One step, I got my stuff. Well, some of you are probably sitting here thinking, well, how do you get it off the column? You get it purified, you put it on there. How do you get it off? This is a very important point. Okay? Very important point. These bindings that we're talking about are not covalent bindings. When we talk about something that's not covalently bound, they're relatively easy to get removed. Let's imagine here that I have this ATP column. How would I get my material off? Well, the easiest way to do it would be to add ATP. The protein, because it's not covalently bound, isn't stuck there 100% of the time. It comes off, it comes on, it comes off, it comes on. Right? If it comes off and now it encounters a free ATP, instead of coming back to the column, it's just going to start shooting through. So I can always wash my proteins off of my column by adding the thing that it binds to. How would I get something off of an anion exchange column? How would I do that? What's that? So an anion exchange column, what's going to stick to an anion exchange column? Positive charge are going to stick to an anion. So, no, actually, negative ions are going to bind, aren't they? We name it for the displaced ion, which means it's going to be positively charged, right? So if I add a bunch of positive charges to this, what's going to happen? I'm asking you a trick question. I can't just add positive charges. I can't just add negative charges. I have to add both because I can't add just ions, right? So I'm going to add salt. I'm going to have positive and negative. My protein's going to come shooting off. Okay. All right. So you see something about how powerful these columns are now for purifying things. Yes?
To wash off the same molecules that you were using before, the DEAE and the other one?
Okay, her question is, that's actually a very good question. Her question is, could I use the supports that are stuck on the column? Kind of like I did with the ATP, right? And the answer is you could. Generally, you wouldn't do that because A, they would be expensive, and B, they might contaminate your protein in a way that you didn't want to have them on there. But it's a very good question. Salt would be much less expensive, and we could always get rid of salt by dialysis. Make sense? Okay, good. Good question. All right, so we now know about affinity chromatography. Let's talk about a more advanced technique called HPLC. How many of you in here have ever run HPLC? Probably several of you have. Yeah, a few. Okay. So HPLC is a technique for separating molecules on the basis of their polarity. Not their charge, but their polarity. Okay? HPLC stands for High Performance Liquid Chromatography. Some people mistakenly call it high pressure liquid chromatography, and that's wrong. Okay? It does operate at high pressure. You'll see why in a minute. Okay? But it's high performance liquid chromatography. All right. How does it work? Well, it turns out that with HPLC, it also works in columns. But instead of having beads about the size of a millimeter, we have little, itty-bitty, teeny-weeny, tiny beads. Hard to see individually with the eye. And these get packed into columns that are typically stainless steel columns. And the reason they're stainless steel is that they're packed together very, very, very tight. That means that if we want to get liquid moving through them, we have to put the liquid at very high pressure, which is why some people call it high pressure, in order to move the liquid through the column. All right? Now, the type of there are two main types of HPLC that I will talk about. Actually, one I'm going to talk about, but there are two that exist. They're called reverse phase and normal phase. Reverse phase is by far the most common one used, and that's what I'm going to talk about here. Okay? Reverse phase chromatography uses these little beads, little tiny beads that are in there, and uses beads that have attached to them very nonpolar molecules. We can think of long carbon chains sticking off of those beads. Nonpolar molecules are on the surface of those beads. Now remember I said this is a technique that separates on the basis of polarity. All right? Water is very polar. Water is not charged, but it's polar because it has hydrogen bonds. Whereas something like hexane is not very polar, no hydrogen bonds. All right? If I take my materials, and usually these are smaller molecules that we're using. We're not always doing proteins here, but usually using smaller molecules, like maybe amino acids. Okay? If I take my mixture of compounds and I apply it to this column and I force them through with this liquid that's going through there, what I will see is that the molecules that will come off first are those that are the most polar. And the molecules that stay on the longest are those that are the least polar, because they'll interact with those nonpolar components. Okay? People use HPLC because it's a very, very reproducible type of technology. Okay? So here's a mixture of 5 compounds that were separated by HPLC. And we can see at 5 minutes, the first compound is coming off. And over here at about 11 minutes or 12 minutes, the 5th compound is coming off. All right? If I take this mixture of 5 things and I run 15 different runs through the column, they will all look essentially identical to this. And more importantly, they'll all come off at exactly the same times that we see them on here. And the reason that happens is because there are so many beads in there, and they're so tightly packed, that those interactions happen consistently as they're traveling through the column. When we thought about that gel exclusion chromatography, okay, we think, well, it could go this way, it could go this way. There were all different kinds of ways it could go. In the case of the HPLC, it doesn't have as many variables in terms of paths that it can take. So HPLC has the advantage that it's very, very reproducible. So I'll repeat that now. The first things that come off in an HPLC column are those that are the most polar. Something coming off at 5 minutes, number 1, is the most polar molecule that's in that mixture. Things that are coming off at 11 minutes over here, number 5, would be the least polar, and these guys in the middle will be somewhere in between in terms of polarity. Yes? This can be used to separate a wide variety of small molecules, including carbohydrates, including lipids, and so forth. Yes? Okay. Well, I'll say it, but we haven't talked about carbohydrates and lipids and so forth. But carbohydrates are molecules that tend to be much more polar than lipids, for example. Okay? So if we were separating here, number 1 would have a higher likelihood of being carbohydrates than number 5, and number 5 would have a higher likelihood of being lipids than number 1 would. Okay? And we can actually, the advantage of HPLC is we can actually separate, usually not say carbohydrates from lipids, but we can separate individual lipids from each other very easily. So in my master's thesis, I worked on vitamin A. And this technique is so powerful that we could separate different forms of vitamin A very readily that were identical chemically, but they had different cis and trans bonds. And those very subtle differences in structure caused them to have differences in polarity. So this reproducibility of HPLC was really, really powerful technique for this kind of separation. That help? Okay, thank you for the question. Okay, let's see. Let's turn our attention to, I want to talk about agarose gel electrophoresis first before I talk about polyacrylamide gel. How many people run agarose gel? Okay, good. Boy, that's really good to see so many hands going up. Agarose gel electrophoresis is one I don't have a good figure for, but I can tell you about it. Right? Agarose gels are, I'll show you what a agarose gel might look like. Polyacrylamide gels schematically look exactly the same. Okay? Although agarose gel sometimes can be done horizontally as a slab instead of vertically, although you can also do vertical agarose gels. What is agarose? Well, agarose is a polysaccharide that has many sugar units attached to each other. So we can imagine it's a polymer. It's something we actually get from seaweed. Right? It's a polymer of these guys. We have a long chain of sugars. Now, if it were just a long chain of sugars, it wouldn't have the property that it does. But these long chains of sugars are interconnected to each other. So I have a chain, connect to a chain, connect to a chain, connect to a chain. All right? Those chains actually make, those crosses between individual strands, make holes. They make holes. And those holes provide a way for things to move through them. We thought of the holes that we had in the beads. We're now thinking of holes at a molecular level, all right, that are between strands of polysaccharides. So these holes act something like a sieve. Okay? Now the holes, I should tell you, are quite large. They're quite large. All right? And the reason that they're quite large is because the things that are moving through them are also quite large. We use agarose gels to separate molecules of nucleic acid, DNA or RNA. And in case you don't know this, the largest molecules in the cell, by a long ways, are DNA and RNA, DNA being the larger of the two. Okay? If you take all the DNA in your cell, in one cell of your body, and you stretch it end to end, 7 feet of DNA. That's how long each DNA molecule is inside of your cells. You have enough DNA in your entire body, if you stretch it all end to end, to go to the sun and back. 180 million miles of DNA. And now you know why you're tired on Monday morning. Right? I got to wake all that DNA up, right? So we're talking about big stuff. Big stuff. Now, typically, we're not separating things that are quite that large. We're separating DNA fragments that are smaller than that. But nonetheless, DNA fragments are pretty large. So we have to have holes that they can pass through. If we block them from passing through, then we're not going to ever get them off of our gel that we're running. All right, so I make a gel. I make my gel that's got this. And now this gel material, which we call it a gel because it's kind of like Jell-O, all right? It has a sort of a liquidy consistency. We set it up so that we have buffer and we have buffer. And we put our DNA molecules that we want to separate right there in what's called a well. Then we close this all up and we apply an electrical current to it. And when we apply an electrical current to it, we make it so that the negative is at the top and the positive is at the bottom. I'll tell you why in a second. Right? What's going to happen is electricity is going to flow and it's going to move from top to bottom, making a circuit. Okay? Now, the reason we put minus at the top is DNA is full of phosphates. There's a phosphate between every nucleotide, and phosphate is negatively charged. DNA is a polymer of negative charge. DNA forms nice rod-like regular structures, and the longer the DNA molecule is that we have, the more positive charges we have. The length is proportional to the positive charge. The length is proportional to the positive charge. That becomes important. The reason it's important is something that has more charges will have more force. Okay? But the force that it will have divided by its size is equal. Right? Larger molecules have more force, that is more negative charges, but the number of charges they have per length is the same. If I have something that has a DNA molecule with 3 nucleotides, it has 2 negative charges, 2 phosphates between them. If I have something that has 6, it has 5. If I have something that has 10, it has 9. It gets longer, I add more positive charges. So the length, negative charges, the length is proportional to the number of negative charges. Now, what does that mean? Well, it means that if I apply a negative charge at the top of this guy, that the force that's acting on each molecule divided by its size is the same. The force divided by the size of each molecule is the same. Okay? I see people nodding their heads. Good. I see other people looking a little more quizzical. Okay? Since the size is proportional to the number of negative charges, and the force is acting on those negative charges, the force divided by the size of the molecule is the same, whether it's a large molecule or a small molecule. So if the force is all the same, what's the basis of separation? This becomes the easy part. If the forces are all the same, the thing that moves the molecules is how fast they can travel through those pores. Small guys can travel faster. Large guys have a harder time getting through. They travel slower. Small versus large. Okay? So, in agarose electrophoresis, the smallest molecules are the fastest moving. The larger molecules are the slowest moving. And this technique is really powerful because I can separate something that has, let's say 500 nucleotides from something that has 600 nucleotides reasonably good. Okay? I can do that separation. All right? And agarose gel electrophoresis allows me to do that. Well, if you understand agarose gel electrophoresis, you're going to understand polyacrylamide gel electrophoresis as well. There's only a couple of considerations for that. All right? Polyacrylamide gel electrophoresis uses a polymer of a monomer called acrylamide. It makes the gel that you actually see right here. And like I said, agarose gels look very similar. The primary difference for our purposes is that polyacrylamide also makes a mesh and leaves holes for things to travel, The difference is the holes are much smaller. The holes are much smaller than they are in agarose gel electrophoresis. Why do we make them with smaller holes? Well, primarily because we use polyacrylamide gel to separate proteins. And proteins are much smaller than DNAs. Proteins are much smaller than DNAs. If we don't make this pore size small enough, we're not going to have a difference between large and small making its way through. Well, there's another consideration for those of you who hopefully are thinking about this. And the other consideration that we have is that proteins don't have a uniform negative charge, right? Some of them are going to be positively charged. Some of them are going to be negatively charged. Some of them are going to be uncharged. And if I put them into this mixture, just like I talked about before, and I put plus at the top, and I put minus at the top, and I put plus at the bottom, and I put my proteins right here, when I turn on the current, the positively charged proteins are going to jump right up here and stay on the top, and the negative ones are going to move, and the zeros are going to sit there and go, I don't know what to do. We're not going to separate things, guys. That's not good. Moreover, we don't have the same phenomenon that we had before where we had the size was proportional to the number of negative charges. That was the key to agarose. DNA, the size was proportional to the number of charges. So in doing separations of proteins on polyacrylamide gel electrophoresis, by the way, polyacrylamide gel electrophoresis people call PAGE, P-A-G-E. And you can, any abbreviation I use in this class, you can use also. Okay? P-A-G-E. If I want to separate proteins by polyacrylamide gel electrophoresis, I have to use a very cool trick. I'm going to describe that trick to you. I have to make the proteins have the same property as DNA had. DNA's property was the longer it was, the more negative charges it had. Remember, DNA is a rod. It's elongated. Most proteins, I said, are what form? Globular. They're not elongated, right? Well, so how am I going to make this elongated, and how am I going to make this have a negative charge that makes it more negative the longer it is? This is the cool trick. You add a substance called sodium dodecyl sulfate, and no, you don't need to know that. You do need to know its acronym, SDS. SDS is a detergent. It is a detergent that when it is mixed with proteins does two things. One, it denatures the protein. I told you last time why soaps and detergents denature proteins. How is it doing that? Disrupting hydrogen, or not disrupting hydrogen bonds, no. Hydrophobic bonds. It's disrupting the hydrophobic bonds, right? And when it does that, it converts it from being globular to stretching out. We've made a rod. Now, the second part's a little harder to understand, but it turns out to be true. And that is the SDS is abundant, and so it coats this rod with SDS. It coats it. And it coats it so well that longer proteins get more SDS, and shorter proteins get less SDS, so that the amount of SDS that a protein has is a function of its length. And guess what? SDS is negatively charged. So now I've converted globular proteins into something that's much more like DNA. It's rod-shaped, and the amount of negative charge it has is proportional to the length of that rod. Pretty cool. So what I've just described to you, just one second, just what I've just described to you is what's called SDS PAGE. SDS applied to these proteins, and then polyacrylamide gel electrophoresis. Yeah, you have a question?
Is the SDS readily removable from the proteins at the end of the process?
Is the SDS readily removable from the proteins at the end of the process? It's a very good question. Yes and no. Do you suppose the proteins at the end of this process would be active? Probably not. Okay? So this technique is usually used as an analytical technique. Remember I said we had those little tubes that came down from the gel exclusion? And I said we use other analytical techniques to see what was in them. We would take a little bit from each of those tubes, and we would put it on each well, and we would say which thing is coming out here, which thing is coming out here, which thing is coming out here. So this would tell me very readily which protein is coming out where, because I would see different sizes on that column. Okay. Yeah, question. Where you at? SDS denatures the protein. That's the key to the way it works. Yes. Well, you wouldn't use SDS to purify it. You wouldn't use the SDS page to purify it. You would use SDS page to analyze it, but it's not going to be a purification. Okay? Good question, though. Yes? I'm sorry, say it again? What about it? The MISO concentration of the detergent? To be honest with you, I don't know the answer to that question. But suffice it to say, you would want to have an excess of SDS in the solution so that you have plenty to coat the proteins. So maybe that answers your question. Okay? Yes? Say it again. For every 2 amino acids, you need 1 SDS. And so that coating is happening on a regular, repeating basis. That's correct.
So since it's not necessarily the length, but the primary structure that determines the protein, if you have one similar length, how far apart can it be really?
Okay, her question is, basically, if you have 2 proteins of similar length, how good is this process at separating those? The answer is pretty darn good. You can separate things with a fairly small distinction in molecular weight. Okay? Yes? Agarose is easier. It's a more liquid thing, but you can do them either. You do it either way. What do you guys say we finish with the song? We've been talking about Henderson Hasselbalch. Let us do some Henderson Hasselbalch. To the tune of My Country, 'Tis of Thee. Henderson Hasselbalch, you put my brain in shock. Oh, woe is me. The PKAs can make me lie in bed awake. They give me really bad headaches. Oh, hear my plea. Salt-acid ratios help keep the pH froze by buffering. They show tenacity, complete audacity, if used with incapacity to maintain things. I know when H's fly, a buffer will defy them actively. Those protons cannot waltz when they get bound to salts. With this the change in pH halts. All praise to thee. Thus now that I've addressed this topic for the test, I've got know-how. The pH I can say equals the pKa in sum with log of S or A. I know it now. All right.